Chapter 10, Part 3, Antimicrobial Resistance. This is the last section in Chapter 10, and we're going to begin this by talking about drug resistance. Your book defines it as an adaptive response in which microorganisms begin to tolerate an amount of drug that would normally be inhibitory. This is a really long, wordy definition. The reality is I can do this in 2 words: former susceptibility. The drug used to work, it no longer does. Now, all drug resistance is due to the genetic versatility and adaptability of the microorganisms. In other words, drug resistance is mainly a genetic component of the organisms. It can be intrinsic as well as acquired, so they can be born with it. It's heritable. Or in some instances, the resistance is acquired. We'll talk about how that happens a little bit later. One thing I would like for you to keep in mind is that chances are any large population of microorganisms is going to have a few organisms in there that are resistant to a drug. Okay? Now, as long as the drug is not present within the population stays the same. The majority of the infection or the majority of that population are susceptible to the drug. It's a non-drug resistant population when it comes to the majority. The difference is if you expose these organisms to the antibiotic, what's going to happen is all of these non-resistant organisms are going to be killed off, and that's going to leave those few resistant organisms to repopulate the area that the previous population was living in, to repopulate your infection site. So this replacement population now becomes the dominant species. You get a lot of drug-resistant organisms. So the organisms did not become resistant because they were exposed to the drug. The drug-resistant organisms were already there. The infection, or the larger portion of the population, becomes the resistant organism in response to the drug being added. So the drug itself did not cause resistance. And I want you to remember that drugs do not cause resistance. Resistance is something that organisms either have or do not have. Exposure to the drug can make resistance more prevalent within the population. There is a difference between the two. So, microorganisms that become resistant, in other words, those that acquire resistance, this is acquired resistance, so they were not born resistant or with a resistance gene. Those that acquire resistance usually do through one of 2 things. A spontaneous mutation within a chromosome. This would be chromosomal drug resistance. Again, it's usually spontaneous, random mutation, changes in the chromosome. Lots of times this leads to a slight change in drug sensitivity or slight resistance, and you can overcome it with larger doses of the drug. New entire genes or sets of genes are usually the result of horizontal gene transfer. Okay? Now, when resistance happens through horizontal transfer, we see things that we term resistance factors. Most commonly, these are plasmids containing resistance genes. Those 3 mechanisms of horizontal resistance horizontal transfer that we've been through previously: conjugation, where we had the bacteria forming a pilus and transferring DNA from one organism to the next. You guys remember this? Okay, pilus. Transformation, which was the uptake of naked DNA, and transduction, which always involved DNA being transitioned or transferred, okay, via a virus. Okay, so viruses here, okay, pili with the conjugation transformation. There's little pieces of naked DNA floating around. Now, these resistance factors like plasmids have got to be there before exposure to the antibiotic or the organism won't survive. The issue becomes that organisms can share resistance genes, and when resistance genes are shared, like via conjugation, drug resistance becomes more widespread in lots of different types of species of bacteria. So, we're going to talk about 5 separate mechanisms of drug resistance. Your book does them in this sort of chart form. I'm going to give them some kind of new names, okay, so that they're easier. These are the names that you'll see on the test. Okay? So I'm going to call them enzyme development, decreased permeability, activation of a drug pump, target alteration, and use of an alternative pathway. We're going to view these as the bacteria hiding from a killer. The unfortunate part is this particular killer is used to being able to do whatever it wants with the bacteria. If it needs to get inside, it gets inside. If it needs to kick down the door, it kicks down the door. This is a particularly dangerous killer. Okay? So, let's talk about the first one, enzyme development. Okay? This is basically creation and utilization of a weapon. Okay? The new enzyme that gets made, and that's always going to be a big key here, new enzymes. The new enzyme that gets made is actually something that will destroy the antibiotic, the killer in this case. Okay? Well, if you kill a killer, he can't kill anybody else. And I know that was a lot of kills. So, if you remember, the active portion of the penicillin molecule is this little guy right here. We called it the beta-lactam ring. Okay? Enzymes like penicillinase, or what are termed beta-lactamases, okay, will actually inactivate penicillin molecules by breaking the beta-lactam ring. Okay? Breaking that beta-lactam ring means the penicillin doesn't work anymore. You've destroyed the antibiotic. So it's obviously no longer going to have an effect. Some bacteria have gotten so good at this that they have actually turned it into an inducible and repressible set of genes. If you remember inducible and repressible enzymes, they're turned on and off as needed. E. coli and S. aureus are great examples of bacteria that have turned penicillinase into inducible and repressible genes. Decreased permeability. Okay? Permeability, you should think about getting past the membrane, getting into the cell. So when we decrease permeability, this is basically like changing the locks to your house. Okay? The majority of antibiotics can get into cells because they can bind to receptors on the surface of the cell. Okay? Binding to that receptor is like putting a key in a lock, and that allows the antibiotic to come in. If these receptors are mutated slightly so that the shape changes, then the antibiotics cannot bind to the receptor, and that different shaped receptor actually prevents the antibiotic from getting into the cell. Now you have to remember, a lot of drugs are going to have to work inside of the cell. An antibiotic that affects the ribosome is going to have to work inside of the cell. There are no ribosomes on the outside of the cell. It doesn't work together. Okay, so this is changing the locks. Uh, here, activation of a drug pump. Right? This is sort of like hiring a bodyguard, so that as soon as the killer sort of breaks into your house and makes its way into your house, your bodyguard is sitting there waiting to kind of kick that individual out. Okay? If these molecules, again, a lot of them need to be inside of the cell to work. If they have to get inside of the cell to work and they're immediately kicked out of the cell, right, then they do not have time to actually work inside of the cell. So it's activation of a drug pump. All right, decrease binding sites. This is what I term target alteration. The most common target that's altered here we see is the ribosome. So I call this target alteration. I think about this like camouflaging, okay? Camouflaging or hiding. What we see is that because the drug itself binds somewhere on the molecule, this can be an enzyme, this can be ribosome, the drug binds specifically to a site on the ribosome. If we change the shape of that site just slightly, right, the drug does not recognize its target. It doesn't recognize its binding site, and the drug never attaches to the target. Okay? We sort of camouflaged or hidden the target site from the drug by changing it just slightly. Okay? So, alteration of a target. Right. The other one, use of an alternative pathway, finding a new way home. As long as you make it home, doesn't really matter what street you go down. So this is kind of detouring away from a dangerous place. Okay? If a pathway gets shut down, like the folic acid synthesis pathway, if it gets shut down by a drug, well, there is another way to get folic acid. We just bring folic acid into the cell. So organisms that are living inside of us are going to be exposed to free folic acid. Right? There's folic acid in our cells. Instead of trying to make folic acid, these organisms can just bring it into the cell. Okay? So this is finding a new way home. Okay? Or you can call it having a new route or a detour. As long as you get there, does it really matter how you get there? Sort of unblocking the pathway. Right. Now, there are a few new approaches out there that are being used. You'll probably see more of them in the future. They're not all that particularly popular right now, some of them. Best examples, RNA interference, or what we call RNAi. This uses small pieces of RNA to actually attach to specific genes in specific places on the genome of a pathogen. And what it does is shut down the ability of that pathogen to make that particular gene. So this inhibits protein synthesis eventually. It pretty much stops the central dogma dead in its tracks. There's no transcription of the gene. There's no translation. Right now, there are drug trials evaluating how well these RNAis work on hepatitis C and respiratory syncytial virus, also known as RSV. So be prepared to see these in the future. The use of bacteriophages, if you remember, these are viruses that specifically attack bacteria. Not approved in the U.S., but we see a few Eastern European countries using this. Most of the reason why we haven't approved it is because it doesn't work particularly well, especially inside of the body. Our immune systems seem to recognize the bacteriophage and eliminate them very, very quickly. But because they're extremely specific to certain bacteria, they are quite useful in getting rid of those bacteria. We just haven't been able to do it inside of the body yet. We see a couple of treatments like Biophage PA used to treat ear infections caused by Pseudomonas aeruginosa. We're seeing some experimentation right now with putting it into wound dressings, especially in combat situations. Adding the bacteriophages in to help keep bacteria out of open wounds. So again, these are on the outside of the body, not necessarily internally. Prebiotics and probiotics, we're much more familiar with these. The probiotics consumed live microorganisms meant to basically supplement or improve the microflora in the intestinal tract. Okay. Helping to kind of augment the organisms that are already there. Prebiotics are basically feeding the organisms appropriately. So getting those organisms there and then making sure they have the appropriate nutrients. Prebiotics do not involve live cultures. Okay? They encourage the growth of certain types of organisms. Now, you can't leave this without talking about the fact that there are interactions between antibiotics and hosts. Selective toxicity is the goal. It's what we strive for. But the reality is that most antibiotics are going to be made with several different types of foreign chemicals, some of which can cause harm to the host. About 5% of all persons taking an antimicrobial drug experience some type of adverse side effect. That's around 1 in 20 patients. The reality is that number should not scare you. An adverse side effect can be anything from diarrhea, upset stomach, a rash, or feeling nauseous, all the way through to things that are very severe like tissue damage or anaphylactic shock. Most commonly we see disruption of normal microflora, so your normal bacteria kind of get out of whack, and allergic reactions. It's rare to see direct damage to tissues where the drug itself actually harms tissues and organs. So, if you remember, okay, your normal microflora colonizes healthy body orifices. Most commonly, we see these in your intestinal tract. This is probably the biggest side effect we see from people taking antibiotics, especially when people take unnecessary or even sometimes necessary broad spectrum antibiotics. The normal sort of beneficial microflora that are there get destroyed along with anything causing an infection. The issue there is that when you destroy those normal flora, you leave room for replacement flora to come in. Remember, those good bacteria were taking up space, okay? Keeping the bad bacteria out. If you wipe out all the good bacteria and it so happens that non-beneficial organisms stay around, they can take over that particular area. Now, there's a specific term for this before I actually move on to the next slide. There's a specific term for this. This invasion of replacement microflora is what we term a superinfection. Good examples of this, yeast infections. Remember, we always talk about the balance between the yeast and the bacteria in the vagina, and how the bacteria keep the yeast in check and the yeast keep the bacteria in check. Well, if the bacteria are killed off with an antibiotic, the yeast will flourish. Okay. Another example here, C. diff, okay. Clostridium difficile, because those bacteria make things like endospores. The endospores can stick around even when the antibiotic is present. When the antibiotic goes away, the endospores will germinate and new C. diff, live bacterium, vegetative bacterium, will begin to multiply and sort of take over. That's what causes C. diff colitis. Okay? So the superinfections can be particularly dangerous. Now, this is the end of chapter 10. Don't get too caught up in memorizing drugs again, or memorizing conditions. Yes, you should know what C. diff is. Yes, you should know what penicillin does. But I don't need you to memorize all the drugs and where they fit. I'm much more interested in you being able to tell me how things work. So, how organisms are resistant, or how drugs actually kill a specific type of bacteria. So, those mechanisms of action and then those mechanisms of resistance.